So paper chromatography we saw last time. We also discussed about how the paper chromatography is a liquid-liquid chromatography, because we have a mixture of uh, solvents there. So for example, last time we discussed that we are using, sorry, why is it hung? Yeah. So we are using butanol, acetic acid, distilled water as a mobile phase in any type of chromatography, and we are going to see partition chromatography, we are going to see adsorption chromatography. We will see that every chromatography is going to have two phases. One of them is the stationary phase and the other one is the mobile phase. So here the mobile phase is the mixture or the solvent system, which is a mixture of butanol, acetic acid, and distilled water. It is in a ratio. It is in a fixed proportion. Is it always butanol, acetic acid, and distilled water? Not necessary. It can be any other. So how do you decide your solvent system, or how you decide which mobile phase is to be used? That depends upon what analyte are you going to separate from the given mixture. Okay, is it only one analyte you are going to separate, or are there some more analytes that you are going to detect and separate? It depends on that. It depends on the solubility of those analytes in that mobile phase. So I have a video here. I'm going to show that, and that will clarify some doubts that you have on paper chromatography, which is a liquid-liquid chromatography. Have you all ever done paper chromatography, or have you all ever seen paper chromatography in your class 10, class 11?
Yes, ma'am.
You have seen, and you all have done also, is
Yes,
it?
ma'am.
But let us just revise. Let us see the method once again.
%99% of people doing job or business.
Hello friends, welcome to the channel solution. In this video, we will show you how to separate and identify unknown amino acid by paper chromatography.
Ma'am, video is not visible.
Video is not visible?
Yes, ma'am.
I am actually sharing the screen. Maybe. Can you see the screen now? What do you see on the screen? On your screen?
Ma'am, the slide of paper chromatography.
Oh, okay. Now, can you see this?
Yes, ma'am.
Yes, so use normal filter paper for this purpose. Cut the filter paper into a strip of rectangular shape. Here we have prepared 3 sample solution of amino acid of volume 10 ml each. Solution A and the solution B is the standard solution of amino acid leucine and valine of concentration 1 mg per ml in distilled water. Third solution is the sample mixture of amino acid which we have prepared for the demonstration purpose of separation by paper chromatography by mixing small volume of solution A and the solution B. Draw a starting line of about 1.5 cm from the bottom of filter paper by the pencil. Draw another line as the end line at the top of filter paper, and this is the line up to which we allow the mobile phase to move. Mark the 3 points by the pencil on the starting line of equally spaced on which we apply the spot of the sample solution. Dip the narrow end of the capillary tube to draw small volume of the standard solution A. Touch the capillary tube gently to the paper to transfer very small volume of the sample on the mark point at one end of the starting line. Again apply the spot of standard solution B in the similar way on the mark point on the starting line using another capillary tube. Again apply the third spot of sample mixture solution of amino acid on the third mark point of the starting line. You can also label each spot below the starting line for its identification. We have a staple thread on the top of the filter paper to hold and support the filter paper in the upright position at the center of the beaker at the time of development. Meanwhile, we have prepared 10 ml mobile phase by mixing N-butanol, glycol acetic acid and water in the ratio 4:1:5 and then allowed the beaker to saturate with the mobile phase paper. Immerse the filter paper vertically in the center of the beaker. Note that the level of mobile phase in the beaker should be such that the spot should not dip into the solvent. Cover the beaker with a petri dish and then allow the mobile phase to move over the filter paper up to the end line through the capillary action. Paper chromatography is based on the principle of partition. Here the component of mixture partition between the two liquid phase. One is the stationary phase, which is the water held in the core of filter paper, and second is the mobile phase. The component in the mixture gets separated due to the difference in the affinity of the component towards the stationary phase and the mobile phase. When the solvent touches the end line, which is at the top of the filter paper, take out the paper from the beaker and mark the solvent front with a pencil, and then allow the filter paper to dry at room temperature. You can see that the spots of amino acids are not visible. So after drying of the filter paper at the room temperature, spray the filter paper with ninhydrin reagent to detect the spots of amino acids. Spray the filter paper.
So if you have amino acids spotted, then there are different reagents that are used to develop. Similarly, like in the olden days, you had those cameras where you would fit a roll in there, and then you click photos and you give it in the photo lab or the digital lab where they develop. So developing is another process. So here also you have to develop. How would you do that? If you have amino acids, you have to spray ninhydrin reagent there.
With a ninhydrin reagent, and after spraying, dry the filter paper in hot air oven at 100 degrees Celsius for about 5 to 10 minutes. Ninhydrin reacts with the amino acids to give bluish violet color. After drying in the hot air oven, you can see the bluish violet spot of amino acids. After this, outline each spot of amino acids on the filter paper by the pencil and mark the center of each spot. After outlining each spot of the amino acid and marking the center of each spot, measure and record the distance of each spot from the starting line to the center of each spot. Also measure the distance run by the solvent from the starting line and then calculate the RF value of each spot. RF value means the retention factor and is calculated by dividing the distance traveled by the individual component from the origin divided by the distance traveled by the solvent.
Okay, I would like you all to just note this down. RF value is equal to distance traveled by solute divided by distance traveled by the solvent. So you have to, after you spray the ninhydrin, you keep it in the oven for drying so that whatever solvent is there and whatever ninhydrin is there, excess of it gets evaporated. The paper is completely dried now. And when you take it out, you will see that the purple color spots have developed. So what do? You first mark the boundaries of the spots. So these are usually oval in shape. Sometimes they have a tail, sometimes they are very crisp. So you have to take note of that, mark the boundaries, and now find out the center of your spot. And from there, you have already drew the initial starting line. From there, you have to measure the distance till the center of your spot. Okay, that is important. Then you have to also measure the distance from your starting line till where up to the solvent was run. So that point where your solvent has stopped running through the filter paper becomes your solvent front. So that is the distance traveled by the solvent. And the ninhydrin spots, the distance that you measure from the starting line to the center of the spot becomes your distance traveled by the solute. So the retention time or the RF value or the retention factor as it is called as is equal to distance traveled by the solute divided by distance traveled by the solvent. So if for example, in this video, distance traveled by the solute is say 2.7 centimeter, which is the first sample that she has loaded. So 2.7 and the solvent front is 7.5. The solvent front is going to be common for all of them for sample number 1, 2, and 3. Okay, so 2.7 by 7.5, you get 0.36 centimeter is the RF value of the first sample. The second sample will be 4.5 divided by 7.5, and the third sample will be 2.9 divided by 7.5. So all these three samples now are having different RF values. So you can also conclude that different amino acids will have different RF values. Okay.
Here we have calculated RF value of spot 1 of standard solution A. Similarly, you can calculate RF value of spot 2 of standard solution B and the RF value of two spots A and B obtained after the separation of mixture sample solution. After the calculation of RF value of each spot, then identify the amino acids of the mixture by comparing the RF value with that of the standard solution. Here the spot A is spot of leucine and B is the spot of valine. Thank you for watching this video.
So that is what we saw just now, that the solvent front is, I hope I have a picture here. Yeah. So this is the solvent front here. If you look at the picture here, this is the solvent front that I am marking. This is still where the solvent has been able to run in the on the paper. Okay. On the paper, now the paper is Whatman filter paper. It has also absorbed, say, water, because our mixture was butanol, acetic acid, and water. So it has, the paper has affinity to absorb water. So water runs through it, the paper becomes wet, and we know now that it is liquid. So the stationary phase also becomes liquid. The mobile phase is also liquid. So hence it becomes liquid chromatography. Now, chromatography, when you talk about chromatography, it is a separation technique. It is now known to us also. There are two types when we talk about chromatography. There is partition chromatography and then there is adsorption chromatography. So today we are going to see both of them in quite a bit of detail. So we will first start with, say, partition chromatography, because we have already seen paper chromatography, which is liquid chromatography. So partition chromatography, one type of partition chromatography is liquid chromatography, and we have already seen that. That's why I will now explain liquid chromatography and the other type of partition chromatography, or you can say, is gas liquid chromatography. Okay. So the principle or the basis of any chromatography is actually based on the partition chromatography. So every analyte that you are going to separate, or it can be a compound, it can be analyte. Analyte, it can be a biomolecule, it can be a drug, it can be a nucleic acid, it can be any chemical compound, because chemistry also, these chromatographic techniques are used. Okay, because I told you that there are a lot of things that are synthesized in a chemical lab also. So some of them are naturally produced by bacteria, by plants. So, but the analogs of them or some similar compounds also can be chemically synthesized. So even chemistry also uses these chromatography techniques as like a biochemist would use it. So any molecular, any molecule, any analyte, any compound will have a tendency to dissolve in a particular liquid. Okay, now as we know that there are two phases in chromatography, we know that there is a stationary phase and there is a mobile phase. So now there is this tendency of this analyte or this compound to have affinity towards either of them. Either it will have more affinity towards the stationary phase, or it will have more affinity towards the mobile phase. So there is a divide, there is a partition, there is a distribution, okay, towards the distribution of the affinity, distribution of the solubility of your analyte, and we are using that property to separate them. Hence, the analyte will distribute between two immiscible phases. What do you mean by immiscible phases? Like they are not easily soluble into each other. So just like, so when we say that sugar is soluble in water. So there is a solid phase, a solid matter, which is sugar, is soluble in a liquid. Okay, so then there the word solubility is used. Miscibility is when two liquids come together, whether they are soluble or whether they are not soluble. For example, water and oil, they are immiscible, and water and milk are miscible. Okay, they can mix into each other very well, they become homogeneous, whereas oil and water cannot become homogeneous, so we can call them as, say, immiscible phases. Now there are these two different phases are there. So immiscible is something which is not soluble, sorry. So now our analyte will either have affinity towards the mobile phase or towards the stationary phase, and the separation is based on this solubility or on this affinity of our analyte. So some molecules and some analytes, molecules or analytes or compounds, drugs, are such that they are more attracted, they have more affinity, they will adhere. Adhere is to bind, is to attach. So they will attach to the stationary phase so tightly that the ones which are not tightly attached to the stationary phase now will come out in the mobile phase. Okay, so if there are two different phases, phase A and phase B, it could be either stationary, this could be, say, your mobile phase. So the value for this coefficient is a constant at a given temperature and is given by the expression concentration in phase A divided by concentration in phase B is equal to your partition coefficient. Okay, so there is this value which is known as partition coefficient, which depends upon the concentration of your analyte in both the phases. If your analyte has more affinity towards, say, phase A, which is the stationary phase, it will remain in the stationary phase. Okay, so the mobile phase will have less of your analyte, the stationary phase will have more of your analyte. So a ratio is going to give you the partition coefficient. Now we will first see how what is the working in the laboratory, how do we perform partition chromatography. So paper chromatography definitely was a liquid chromatography, but now we will also move to column chromatography, something which is known as column chromatography, which is regularly used on a small scale, on a big scale in the laboratories to separate certain molecules, certain compounds. Okay, so today we are going to see these two types in detail. Now we will go to, say, a blank slide first, and then can you see this?
No, ma'am.
Okay. I guess now can you see it?
Yes, ma'am.
Yeah, okay. So there are, I told you, the solute molecules, they have affinity, different affinities between two liquids. So here, what is the, your stationary phase is also going to be a liquid, and your, say, mobile phase is also a liquid. Now your analyte that you're going to separate, I give you a mixture. Okay, if I give you a mixture of your microbial extract, so now you have grown some bacteria which is producing, say, very useful compounds which you want to provide to the industry, and you grow it in the laboratory. Then what I told you is you have to separate the bacteria from your medium. So what do you do from your liquid medium? You centrifuge, you remove the cell mass. Now see, there are two types of processes. One, if the bacteria is producing, suppose this is your bacteria, and now this bacteria is known to produce, say, some very useful compound or some very useful enzyme. So let us say that this is, say, lipase. Okay, it's a lipase enzyme. Now there are two things: either the bacteria is going to secrete the enzyme and keep it inside, either or the bacteria is going to release the compound outside into the media. So for example, again, if you have your flask, if this is your flask, which has media, and then if these are the bacterial cells inside your media, so let us consider these. So here, if they are synthesizing lipase, if they are producing lipase inside, and this is the lipase that the industry wants, okay, or this is that very important compound that the industry wants, and you are working on it. Now, how will, if it stays inside, there are different ways of processing it. If it does not stay inside, and after say 72 hours, the cell breaks, and then it gives out the enzyme. So your enzyme is now going to be in the liquid medium. Not necessarily the cell may break after 72 hours, the cell may also die, it reaches to a stationary phase. At that time, all the nutrients in your medium are exhausted, are already used by the cells to produce the lipase to grow in those 72 hours. So the cells practically have nothing to feed on, they may die. But they may also release the enzyme outside. So such secretions are called as extracellular. So lipase becomes an extracellular enzyme. Now, if your enzyme is there in the medium now, so it becomes very easy for you to separate them. How? You just have to do centrifugation. So when you do centrifugation, you're going to spin your sample at a very high speed. All the cells will settle at the bottom. Already they have given out the enzyme in the liquid. Now what you do, you take your mixture, you take your medium, which is now clear, and that is going to have your lipase. There are cases where the cells may die, but may not give out whatever compound they are forming, whether it is lipase or drug or a very important molecule or something, which is still there inside. Now, in such a case, it becomes difficult for you to extract that useful compound. How? Because even if you centrifuge, the cell mass will settle at the bottom, but your extract, which is there as a supernatant, will not have any of them because the cells have kept all those inside them. So these, the ones which are not given out, and the compounds which are staying inside the cell, that process or that phenomenon is intracellular. So these are now known as intracellular enzymes or intracellular substances. So some of them are extracellular, which are thrown out of the cell. Some of them are intracellular, which are kept inside the cell. So in that case, you will have to take a cell pellet and you have to lyse the cells later. That is another treatment that you have to give. So now if I give you a mixture, say of an extracellular enzyme, and then I tell you that now you have to separate them, then how would you do it? Either you would use a chromatographic technique, that is what we are learning. So separation technique. Now the cell is not only producing lipase, because the cell has so many other genes also. So it might produce, say, a mixture of lipase, protease, amylase or whatever. So it might not only produce A, but it will produce A plus B plus D. So your extract or your supernatant that you have got on centrifugation will not only contain the molecule of your interest, but it will also contain B plus C plus D, which you do not want. Okay, which you do not want. Now, how would you separate? Because hardly we can see them. If you physically, if you would have been able to see them, you would have put your forceps or something and scooped it out. But that is not possible. These are invisible things. So now you will have to use a chromatographic separation technique, so that first you also know that your compound is there. Secondly, you will be able to separate them from each other. So now you will now understand that why these two phases play an important role. So you can choose your mobile phase based on what you have to separate. If you have to separate A, then your mobile phase depends on the properties of that particular compound A. Okay, so your mobile phase has to be dependent on the substance that you have to separate. The stationary phase also depends on the type of the compound that you have to separate. So now separation of your analyte will happen due to differences in partition coefficients. Okay, we just saw what is partition coefficient, because I told you that analyte will have the affinity in a different way towards your stationary phase also and towards your mobile phase. It may be having a strong affinity towards the stationary phase, so it will retain on the stationary phase or with the stationary phase, and the mobile phase will have less of it. So then that solubility, that affinity difference is known as the partition coefficient. So basically, when you talk about partition coefficient, you will say that these are relative solubilities. Why relative? Because you are going to compare, no? You will say that this analyte A is more soluble in the mobile phase as compared to the stationary phase, or the compound B is more soluble in the stationary phase than the mobile phase. So you're going to compare both of them. So that's a relative solubility, and the stationary phase is a liquid. Now, when we are talking about partition chromatography, the stationary phase in partition chromatography is always liquid, okay, which is held in place. Either you coat it on some solid support or you form chemical bond with solid support and then you pack it. So what does it mean now? So suppose if you have a solid support, this is your solid support. It has a particular shape. So this is your solid support. Now your stationary phase is going to be coated over it like this. So this is your stationary phase. The red part is the stationary phase and the green part is your solid support. So now where do you place it basically? So we are talking about column chromatography. So the column basically looks like this. Have you all seen burettes during your chemistry practical in class 12?
Yes, ma'am.
Yes. So the burettes are like this. So we are going to use something which is very similar. This is the column. Okay. So you're going to pack these stationary phases. This is just one particle of your stationary phase. Now your stationary phase is going to be present in many numbers. So your column is actually going to be full with lot of such particles. Okay. And then the solid support is just going to be here inside somewhere like this. So this becomes your stationary phase. Now what you do? Coating is mostly, done physically. So just before you are packing your column, you can just coat your stationary phase, or there are now readily available, commercially also available coatings are available. So you can order them and you can pack them, or then you can also have chemically bonded coating, which are much firmer. So when you do physical bonding, the bond is not very strong. So the coating may come off, but when you have a chemically bonded coating, then that kind of bonding is very strong and it can be reused also for the next column also. Now, when you pack this with your stationary phase, you will have to now add your mobile phase. So see, this is something very different from the paper chromatography. Even in the paper chromatography, there is ascending and descending paper chromatography. Okay. What we saw in the video a few minutes ago, what was that type? Was it ascending or was it descending? We saw some video just now. Was the mobile phase moving from down to up, or was it coming from up to down?
Ma'am, down to
Yeah,
up.
it was moving upwards. But in column chromatography, what you will see is the mobile phase always moves downward. Okay, the mobile phase is always going to move downward because of the gravitational pull. So all the column chromatographies that you are going to see, you will see the same thing, that the mobile phase is poured from the top and it will always move towards the bottom. Okay, so now this is your column. You have packed it, say, with your stationary phase, and now you are pouring the mobile phase also, which will now move through the column. So that is like first time you just run your mobile phase for the first run. After your stationary phase is now become wet, you will now load your sample. So what will you do? You will load the mixture that has been given to you. So the mixture that has your analyte. So you will pour it on the top of the stationary phase. Okay. Now this mixture can be A plus B plus C. Okay. Or just to avoid the confusion of having too many molecules, let us say A plus B, out of which you are interested only in A. Now you have loaded it. The mobile phase is full with buffer. Okay. When you add the mobile phase is a liquid thing. So it will go and cover all the gaps also. It will run between all the gaps between the stationary phase. Okay. The stationary phase is tightly packed. The particles are very close to each other. I haven't drawn that detailed diagram here, just for you to see that the mobile phase is actually able to run through the column. Now, before you actually pack the column, the most important thing is to have a barricade here. What will happen if you don't have a blockage, or if you don't have a stopping here? What will happen? If I don't have a plug here, what is going to happen? Any idea? Yes. What will happen if I don't have a plug? If I don't have any barricade here, this particular part? What does a plug do? You have done some microbial experiments. So you put a plug on the flask. What is the purpose of that plug on the flask?
To prevent it from getting contaminated.
Yes, correct. So it means it is avoiding the entry of the bacteria. Okay. So in this case, we are not bothered about the bacteria, but if we don't plug it, what will happen? Whatever I pour from the top will come down as it is. Okay. And then what is the use of packing the column then? There is no use of packing the column also. So you always have to have a plug. And you will say that, ma'am, there is a stopper here, then why do we need a plug? If I always have locked the column, then nothing will drain out. Also because we are having stationary phase, we are having a mobile phase, and then we are having our sample also. So the solubility now, your analyte, if it is more soluble in the stationary phase, if it is more soluble or has more affinity to the stationary phase, will retain here for a longer time. And the analyte, which has more affinity to the mobile phase, will come down. So this kind of acts like a plug, it acts like a filter, both. And then the mobile phase is going to trickle down and you're going to collect it from the bottom. So the plugging of a column is very important. You either use a cotton plug or you either use a glass wool, because the stationary phase will also, it's so tiny, it's so small that it might just come out. And if your stationary phase, that is not the ideal way a column has to be packed. Before packing a column, what are the few important things that you have to do is choose a proper column. Choose a proper column. What do you mean by choose a proper column? Depending upon how much is your sample that you're going to load, depending upon all of these conditions, you have to choose a proper length of the column. Then you have to plug the column. Now this plugging of column can be done by cotton plug, or it can be done using a glass wool also. Okay. Then these are the two first important things that you have to follow when you pack the column. There are other things is like, you then have to pack your stationary phase. Now packing of stationary phase is again can be done in two different ways. Either you do the dry packing or either you do the wet packing. So what do you mean by dry packing? You simply, so this, whatever is your stationary phase, it could be silica, it could be any other polymer like starch. Okay, so your stationary phase, it is available, say commercially, you order it, you then pour the dry powder, and then on top of that you add your mobile phase. Okay, that is known as dry packing. And what is wet packing? You first soak your, so we are going to call this stationary phase as, say, so soak your stationary phase. Let us use the word stationary phase. And then in the solvent and then pack it. Okay, so you have a cotton plug, you have the stationary phase therein now, which is coated, say, on a solid support, and then you pour your mobile phase, and then you are now loading your sample on it. So when you load your sample, the column is now ready. Now what will happen? Now some solute particles are more soluble in your stationary phase, and some solute particles are less soluble. So the ones which are more soluble and the ones which are more attracted will sit or will remain in the stationary phase, and the ones which are less soluble in your stationary phase, for example, say particle B, we'll give it like a shape of, say, a star. So if that is not finding any attraction to the stationary phase, will just come out as it is. Okay, and then you may be able to collect it. So depending upon, so suppose if B is, if you know that my analyte B is more soluble in the mobile phase, and I can separate it in this way, indirect separation, I will add A plus B to it. A will remain on in the stationary phase and B will come out. So now I have received a pure sample. Okay, I have received my pure analyte, and the one which is retained on the stationary phase anyway is of no use to me. So I will not, I'll not bother about it. Okay, so now here if your analyte is more attracted or has more affinity, or solubility to the stationary phase, will it come early out or will it come late outside? I mean, will it pass the column quickly or will it pass the column very late? What will happen if your analyte has more affinity or solubility in the stationary phase? Will it come out first or will it come out last? Yes, you can write in the chat box, all of you, please. If your analyte has more solubility or more affinity to the stationary phase, will it remain in the stationary phase and come late, or will it leave the stationary phase and come out quickly? I'm waiting for your answers in the chat box.
Ma'am, it will come quickly.
If it has affinity towards the stationary phase, so what will happen? It will adhere to the stationary phase. So will it leave the stationary phase quickly?
No, ma'am.
No. So it will, correct,
No.
it will go slowly. So if depending upon your analyte, now if your analyte has more attraction towards the stationary phase, it will come late. Okay, it will come late. If your analyte has no attraction towards the stationary phase but has more solubility in the mobile phase, then it will come out quickly in the mobile phase. Did you understand this concept?
Yes.
Okay, not only Anushka, others also. Have you all understood what I'm saying? See, this is very important. These are very basic things and you all should know. Now, if you talk about the applications of partition chromatography, it is separation and identification. I'm going to dictate this. It'll be great if you all can write it. Okay. Separation and identification of amino acids, tannins, carbohydrates, glycosides. These are all biomolecules which are very important. Then you can determine, it is also used, this kind of chromatography is also used, partition chromatography is also used to determine the quality of water. You can also use it in, say, identification. So if you have a mixture, typically you can use this technique to separate your analytes from the mixture. So you can identify, you can analyze petroleum products also, because we know that it's a mixture. Fractional distillation also is done of this to attain a lot of important intermediates. Okay, so similarly we can also use, say, chromatography to do that. And then analysis of pharma products. Intermediates. So these are the important applications. Advantages are also there of partition chromatography. You can actually separate any type of mixture here. Okay. Advantages is separation can be achieved. Second advantage is that there is like a wide choice, or there is a wide range of mobile phase that you can choose. So wide choice selection of mobile phase, because see, mobile phase is very important. Both the phases are very important rather, but since we are working on the property of partition chromatography, mobile phase is something that is going to run through your stationary phase. It keeps on running. So there is a wide choice of mobile phase. You can alter the mobile phases, because like there are certain analytes which will be more soluble in, say, solvent A, but their solubility is also good in solvent B, solvent C. So depending upon your analyte, so A plus B, A plus C, A plus D. Now A plus B, so this A is my analyte. Okay, it is also soluble in ethanol. Say it is also soluble in acetic acid, and it is also soluble in water. Okay, for example, this is just an example. Now the other analytes B, C, and D, their solubility is different. So now if I know that my particular analyte is soluble in all the three, then I can really do a lot of variations. So if suppose A is soluble in amino acid, but B is not soluble in not amino acid, acetic acid, and B is not soluble in acetic acid, but B is soluble in ethanol. So I cannot use ethanol because both of them have solubility in ethanol. So I will avoid ethanol and I will go for amino acid, because I know that A is soluble in amino acid, in acetic acid. I'm very sorry. A is soluble in acetic acid, but B is not soluble in acetic acid. So I can decide, I can choose, I can make combinations and make my mobile phase. Okay. Then you can also have atomized chromatography. You can also have atomized partition chromatography. There is manual chromatography that you can always do in the laboratory. But then the disadvantages are that it is very time consuming. One has to be really very patient doing this. Okay. And automation is very expensive. So it is very time consuming. It will require more mobile phase. Automation techniques are more expensive. And also sometimes reproducibility may vary from person to person, whoever is performing it. So if you perform, and if the other student in the class performs, you may get the same procedure. If you follow the same procedure, you may get different results. So reproducibility is also sometimes questionable when you go for manual way. Okay, but and if you want to go for automation is very expensive again. Have you all understood this? So we move to adsorption chromatography. Is there any doubt here? Is there any doubt? Okay.
No, ma'am.
No. Okay. So we saw chromatography are of two types: partition, adsorption. We saw what is partition chromatography. Now we will move to adsorption chromatography. This is also a column chromatography technique that we are going to discuss today. It's a simple chromatography. It's a simple column chromatography. Again, we are going to use the column which looks like the burette that you used in your titrations in chemistry practicals. It's a simple column chromatography. Now, the word, what is the difference between adsorption and absorption? Please write it in the chat box. What is the difference between the word adsorption and absorption?
Ma'am, adsorption is only surface
And
and
what happens?
absorption is full.
Okay, so it means that like adsorption is a surface phenomenon. So whatever so there is this property of a compound or a molecule attaching to on the surface of other substance. Okay, so that's a physical property. When you say absorption of water happens where the particular molecule, it goes inside and it interacts with the material inside, it stays inside or it gets modified there. But here the physical bonding happens between the surface of one molecule and the surface of the other molecule. So this involves the adsorption of solute molecules on a stationary phase. Now, if I have an analyte, so what will happen? The analyte is the one that you want to separate. Okay, the analyte molecules will adsorb on stationary phase. So it's a physical property. This in this type of chromatography, separation of sample mixture occurs due to differences in the adsorption affinities. When we were talking about partition chromatography, we were talking about solubility. Okay, we were not talking about adsorption there. We were talking about two different phases which are liquid, but which are immiscible phases. Okay, which are not going to mix into each other. They are immiscible phases. So we are talking about the partition. The analyte will be more soluble in the stationary phase, will be less soluble in the mobile phase, and vice versa. Here in adsorption, we are not talking about solubility affinities, but we are talking about adsorption affinities. Okay, we're talking about the adsorption affinities. So if you are loading your sample mixture, which has your analyte, now this analyte may have more affinity towards the stationary phase. So it will go and sit, it will go and attach to the stationary phase. Okay. So adsorption affinities of sample mixture. So this is the property that we are going to exploit. Now, mixture to be separated, what happens is you are given a mixture and that contains your analyte or analytes that need to be separated. For that, again, you need a suitable or appropriate solvent system. Okay, again, what do? You again have a column here. So you choose a proper column, plug it. That is very important. So plugging is again very, very important. So you're going to have filled something in this. Your filling is going to be your stationary phase inside. So So this yellow thing is your stationary phase. Somebody's mic is on. Can you please put off? Okay, so this is your stationary phase. Now you will, again, the packing of the stationary phase, as we discussed, can be done by dry method or can be done by wet method. You know what is dry now, you know what is wet. We will also discuss it more. Okay, so suppose now you have packed the column. What will you do now? You're going to load your mixture on the top. So this is my mixture. And this mixture has the analyte that is to be separated. This is a mix. Again, let us say, take a name. Let's say A plus B. Okay, A plus B is my mixture. Now this particular yellow color filling is the stationary phase. Okay, it's a stationary phase. I can also call this as my adsorbent now. Why adsorbent? Because whatever I am going to load is going to get absorbed on the stationary phase. Not all of it, but partly. Okay, so you pack the column, then you load the mixture. Now what will you do after loading the mixture? You have either you have opted for dry or either you have opted for wet. So if you go for dry, what you do, you first pour in your stationary phase and then add the mobile phase over it. Okay, if you go for wet, you soak the stationary phase in your mobile phase first and then you pack it. After you pack it, you load the mixture on your stationary phase. Now your mobile phase will run through the column. Okay, it will run through the column. So if you suppose this is your mobile phase. Now the red part is the mobile phase. Okay, this is going to run through the column. Of course, because it's liquid, it will find gaps and it will trickle down. Okay, it will be able to run down through the column. So now as the mobile phase passes through your mixture, the mixture contains, say, this and this. A part of it will be more will have more affinity towards the mobile phase. A part of this mixture will have more affinity towards the stationary phase. So it will depend now based on their adsorption affinities, which will move first and which will move last. Okay, so the substance, the mixture or the analyte which has more affinity for stationary phase will be slow in movement, and the substance or the compound which will have affinity with the mobile phase will move fast and will reach the end of the column very fast. Okay, so if now my mix has separated, suppose, so it will move down and A plus B will now split into A and B. So what will happen is somewhere here the B has moved down. Okay, now this is my analyte B. A has also moved down a little bit. This time it is of different color because A plus B was giving me green color, but now B has separated, it has become blue in color. And I will choose, say, something like this. This is my A analyte. This is A, this is B. So B is moving faster. It is moving faster towards the end of the column. A is still there in the stationary phase. Now my mix का thickness इतना ही नहीं रहेगा, मेरा mix का thickness कम हो जाएगा. Okay. So suppose it will reduce because it is separating also. As it is moving down, as the mobile phase is also moving down, the sample will also move down. See, I will have to draw another column again to show that the sample has moved down. Instead of that, I will increase the length of the column to show you all that yes, the sample is moving down. So जो thickness है मेरे mix का जो मैंने load किया है, whatever I have loaded is going to reduce because separation is happening already. The B component has moved down because it has more affinity towards the mobile phase. The A component has stayed back on the stationary phase because it has more affinity towards the stationary phase. And in this way, the B will come down and then it will trickle down and you can collect your analyte. Okay, so this kind of separation you will see is known as distinct separation. Okay, so as your substance, as your analyte moves, first, how was it? First, how was it? It was just a band of green color. This was the mixture that you loaded on the column. So I'm going to show you only the mixture. Now focus. Okay, concentrate on this. Now you have added the mobile phase. The mobile phase will pass through it. Then in the next 2-3 stages, what is going to happen? A little separation you are going to see, and you will see like this. This is my A analyte, and this is the B analyte. This is, say, in the second phase of separation. Can I say that this is distinct separation of A and B? Can I say that the second one here is the distinct separation of A and B? This is A and this is B. Are they separated completely, distinctly? Can I say? You can answer in the
Yes.
chat box. Are they separated distinctly? Can you nicely separate them? They still share a border between both the analytes. There is a common border. So is this a distinct separation? No, it is not a distinct separation. So what is a distinct separation? Now a distinct separation is going to be like this. So your analyte A will be here. In between that is going to be your stationary phase. Okay, imagine that this is happening in the column. And then your B. Now is this distinct separation the third picture? Is this distinct separation? What is distinct? Like two bands are separate. Are they separate? This is A and this is B and this is your stationary phase. Now are they separated properly?
Yes.
Yes. What about others? Can you all please type yes or no? What do you think? Yes, they are distinctly separated. Now also tell me that depending upon the position of the band, the A is on the top, the stationary phase is in between, and the B analyte is down, which is moving towards the end of the column. Whether A has more affinity or whether B has more affinity to the mobile phase? What are the affinities? Okay, tell me like this. What is the affinity of A towards and who is B attracted towards? Whether A is attracted to stationary phase or mobile phase, or whether B is attracted to stationary phase or mobile phase. Y'all can type the answer in the chat box. What are the affinities of A and B? Juhi, B is attracted to stationary phase. Then if it was attracted to stationary phase, it should have stayed up with the stationary phase only. Why is it going down? Why is it going down to the end of the column? Because it is attracted towards the mobile phase. Juhi, did you understand this? Juhi, did you understand what I said just now? Okay, good. So A has more affinity. It will stay adsorbed on the stationary phase. B has more affinity to the mobile phase, so it will be adsorbed on the mobile phase and it will come out. So now if you look at this colorful picture here, it looks like a chromatogram. It looks like a chromatogram. So different bands. Okay, these you can address them as bands. It's a chromatogram now because you have obtained distinct bands on the stationary phase based on their different affinities. Okay, based on the different affinities. So what will happen now after you develop a chromatogram? The now column is washed. Okay, because they can't stay like this forever. So you keep on washing. What do you mean by washing of column? You keep on adding more and more mobile phase. So this B band will keep on coming down. Okay, it cannot stay there forever like that because you are interested in harvesting it. You are interested in, say, so it will come down with the passage of mobile phase. It will keep on coming down and then it will come here and finally it will elute. Okay, and you will be able to extract it, or if so, so it has to be eluted. So more and more washes are given, and since B has more affinity to mobile phase, the B component will come out in the mobile phase, and now you will have your pure component or pure analyte. Okay, the fractions will be collected in different vials. So you cannot see when you do it in the laboratory, not necessarily you are going to see these colorful bands that you're seeing it right now, not necessarily. So then there you have to analyze them also. So what would you do? You will collect the fractions and then you will do a UV spectroscopy, UV visible spectroscopy again. So when you do a UV visible spectroscopy, we now know that the light interacts with the different particles. These particles can only absorb light of a particular wavelength. So you have to set the wavelength of the UV visible spectroscopy first, depending upon what analyte you are expecting. If this analyte can only be seen, say, at 290, not seen, can be analyzed at 290 nanometer, then you will have to set the wavelength at 290 nanometer, which is a UV, and then you will get some reading. And if you get a satisfactory reading, it means that your analyte is there in the fractions. Okay, so now when we see how we go about the stationary and the mobile phase, choosing of stationary phase is also important. Now what does your stationary phase, choice of stationary phase depend upon? It depends upon the nature of your analyte, or you can also call it as solute. Okay, mixture, because whatever mixture you are loading, it can be a mixture of enzymes, it can be a mixture of enzyme plus amino acids, drugs plus nucleic acid, whatever mixture you are loading, it can be of various things. So whatever you load on the stationary phase will depend on your solute mixture. Just like you decide to wear a dress depending upon the function that you are attending. If you're going for a wedding, you're going to be dressed appropriate for a wedding, and similarly for other events also. Similarly, depending upon the solute mixture, the stationary phase will be chosen. There are some common adsorbents which are used, like alumina, then there is silica gel. Okay, then there are some, say, natural adsorbents also, like starch. Then in this also there is sodium carbonate, calcium carbonate. These are also naturally found. So inulin is there. These are some common adsorbents. Now, most commonly used is the silica gel. It is used as most commonly is the silica gel. So if silica gel, what is silica gel? It is basically hydrophilic in nature. Okay, hydrophilic is it has more affinity towards water. Why? Because it has this particular group which is known as SiOH. This is the functional group there, and this is known as selenol in silica. So this is making the silica gel more hydrophilic. Now this selenol group, this SiOH group, can interact chemically with various reagents. Okay, so have you heard about HPLC? High performance liquid chromatography. Have you heard about HPLC?
No.
No? Okay. So in HPLC, you have a column packed. This column is the silica column. And what happens? The mobile phase passes through the column like this. Okay. So before you pass your mobile phase, there is a small, or say a port from where you can inject your mixture, where you can inject your sample. So sample will be first loaded here. Okay. Now your sample is going to be a mixture of lot of different things like yes, अलग-अलग चीजें होंगी उसमें. So now when you insert your mobile phase, the same principle, okay? Some of them will have affinity towards the mobile phase, some will stay on the stationary phase. Now suppose the blue dots have more affinity towards your mobile phase, they will move faster. With the mobile phase only they will move, and then you will collect the eluent from somewhere and you'll go for spectroscopic methods for analyzing it. If the red has the second most affinity to the mobile phase, the red will always be after the blue, because blue ka sabse zyada affinity hai mobile phase se, followed by red. The green one says I do not like the mobile phase, I want to stay on the stationary phase only. It will then be retained on the stationary phase. So depending upon the adsorption affinities, your separation occurs in HPLC. Here also it happens the same. So the silica which is packed in the column, that silica will have selenol groups. It will interact chemically with various reagents. It will also interact with and it will form a bond. Okay, it will form a bond and it will convert the hydrophilic silica into hydrophobic. Okay, because it will now interact with the various functional group on the reagents and it will turn from hydrophilic to hydrophobic. Okay, now when you talk about silica, why am I telling you all this about this? Because HPLC is also used in separation of important bacterial fungal intermediates. It is also used in separation of biomolecules, proteins. Okay, a lot of things are separated. Drugs, in pharmaceuticals, HPLC has a lot of importance, significance. So that's why I'm telling you this. So in HPLC, you have a silica column. Okay, so we saw that we pack a column like this in a laboratory. Now just make it horizontal. उसका नीचे का ये जो टिप है, अगर आप इसको काट दोगे, यहाँ इसको सील कर दोगे, यहाँ इसको सील कर दो और उसको हॉरिजॉन्टल कर दो या वर्टिकल रख दो, it becomes a column. Now it has a slight opening here which is attached to a small pipe, and here you can inject your sample and you can inject your mobile phase. So that is how an HPLC works. It's basically just like a column chromatography, but it is a more sophisticated way. Glass column chromatography is the outer container is glass. In the silica column, which is packed in an HPLC, this is a metal, this is a hard metal covering, and this silica can be reused. मतलब एक बार use करो, उसको अच्छे से wash करो, make sure that all of your analytes are washed off, then it can be used for the second round also. So silica columns go for years. Okay, and then after they get clogged, it is just like our water filter, AquaGuard. We change the column, we change the filters once in a year क्योंकि वो clog हो जाते हैं. Similarly, the silica column also needs to be serviced, it needs to be cleaned, it needs to be changed. Now on the silica, I told about the selenol groups. Now the selenol groups are either bound, they are either free, or they are, say, reactive. So in this is how SiOH, okay, this is the silanol group. Sometimes this is bound, sometimes it is free. This is the reactive silanol. Okay, so here you will see less activity. So if it is bound, then the activity is less. Okay, here if the free silanol groups, the number of free silanol groups is more, the activity is more. So the silica gel is actually very, very active. If the reactive silanol gel numbers are also more, then the activity of silica is more. Okay. Selection of mobile phase. Now this was about stationary phase. You can use silica, which is very commonly used. Basically, silica is ideal, I would say. Very, very commonly used, very ideal because of its nature, because of the chemistry that it has. Now we go to the mobile phase. So mobile phase, the selection is also based on the strength of the solvent. The strength of the solvent. This strength of the solvent is responsible for the partition coefficient, because any type of chromatography you're going to see is working on the principle of partition chromatography. Okay, partition coefficient, sorry. So solvent strength is also the solvent that you choose is based on the strength of the solvent, and that is indirectly or directly connected with the partition coefficient. And secondly, the polarity of solute mixture. Okay, the solute mix that is to be separated. Okay, now there are different mobile phases that one can use. Acetonitrile is one of the mobile phases. Then you can see there is methanol. H2O is also one of the mobile phases, because biomolecules, when you talk about biomolecules, the solubility of biomolecules is more they have more affinity towards water. So yes, H2O can also be a mobile phase. Okay. So this is all about adsorption chromatography. Now, which is the one type of adsorption chromatography? Example or a type of adsorption chromatography is gas solid chromatography. Okay, it is also known as GSC. The gas here is the mobile phase and the solid is the liquid, the stationary phase. Okay. Again, when we come to the packing of column, so when we come to the packing of column, you need a proper size of the column depending upon the material, depending upon what you want to separate. Then cotton plug, so plug the column, glass wool or a cotton plug. Again, packing of column. So packing of column can be dry or wet. Okay. So now after that, there are some points to be remembered. When you pack a column dry, okay, when you do a dry packing, there are certain disadvantages. First disadvantage is that, see, what are you going to do? You're going to probably pour the silica in the first, you're going to pour the silica powder in your column. So it's basically, if you have felt rangoli in Marathi, रंगोली का जो टेक्सचर होता है ना, वैसा ही सिलिका का भी टेक्सचर होता है, ड्राई सिलिका का. इट इज वेरी फ्री फ्लोइंग, पाउडरी, बहुत ज्यादा टाइनी होता है. सो इफ यू fill the powder inside there, along with the powder, there will be air pockets also, okay, that you are going to include. Now you add your mobile phase, so your silica will get wet, but it will have air pockets, okay. Now it is very important that you remove those air pockets. So for the fourth point is removal of air pockets. So when you do a dry packing, the first drawback is more mobile phase is required, because since there is no the silica is not wet initially. So when you add water onto something which is dry, it will tend to take more mobile phase. That is also one of the most disadvantages. Why? Because we are using acetonitrile, we are using solvents which are expensive basically. So when you're using such things, you need to have a control on how much you are using. Then it is not preferred. Dry packing is not preferred, whereas wet packing is most preferred. Okay, it is most preferred. It requires less solvent also, or less mobile phase also. Because what you have done for wet packing, you have already mixed the adsorbent previously before packing the column. Overnight usko soak karke rakha hai mobile phase ke sath. So it reaches an equilibrium stage and then you pack it. In both the cases, there is chances of air pockets getting into the column. So it is very important to tap the column. So how will you remove the air pockets? By tapping the column. So because when you tap the column, the air pockets are going to come up and escape. Okay. So when you fill the column, tap it and remove the air, they come out in the form of a bubble. Now the column should always be, column should never be dry. Rather than that, I will put it like this: column should not be dry ever. Okay. So when you pack it, even if it is moist, you need to make sure that your mobile phase is always there. One layer of mobile phase is always there. Okay. Why? Because if you let it dry, this is silica, it will crack. And if it cracks, it is not an ideal column. It is not an ideal column. So there will be crack in your column, and that is not going to give you a proper separation also. That kind of column is not allowed. Okay. Now the sample will be poured from the top, and this pouring of sample can also be called as loading of sample. This pouring of sample can also be called as loading of sample. And then when you run the mobile phase, then pour or pass the mobile phase. When you pass the mobile phase, a chromatogram will develop. So this is how you do adsorption chromatography. Now, are there any doubts? I will move on to applications, advantages and disadvantages. Any doubts? कुछ समझा है, नहीं समझा है? I'll take it as you have understood everything. Applications are: first, separation and purification of nucleic acid. Na, that is the nucleic acid. You can also separation, say, of high molecular weight compounds. Separation of aliphatic and aromatic compounds. Such as lipids. So again, these are biomolecules we are talking about. Fourth, you can analyze or you can detect plant pigments or fat soluble vitamins. Okay. Advantages are, again, a wide range of mobile phases. So you can say wide choice of mobile phase for separation. Okay. Second, complex sample mix can be readily separated. So you can use a complicated sample mix also, not just A plus B, but you can have A plus B plus C plus D plus E, complex sample mix. And not only the number of analytes, more number of analytes does not make it complex. Sometimes the chemical nature of that particular analyte that you are separating also adds to the complexity. The affinity towards adsorption also adds to the complexity. The third, complex sample mix can be separated. The third one is, the most important is simple and cost efficient, because industry, when you look at a larger scale, the process, everything right from the production to the purification to detection, everything adds to the cost. So whatever material we are actually buying from the market, all of the costs are added to it. So how was it manufactured, how are they packaging it? So we pay for everything, we just don't pay for one process. So basically this step is also important. But here you see the detection separation is simple and it is cost effective. Manual and automation, both can be done. Automized adsorption chromatographies are also available. You can even do it manually also. Now again, the disadvantages is also similar to the partition. Reproducibility is a big disadvantage. The results are not reproducible. Okay, now some molecules and compounds, no, because we are working on the principle of adsorption, some molecules and compounds have a high affinity, so they can, molecules and compound can permanently adhere is to bind, permanently bind to the stationary phase. So it becomes very difficult to remove such compounds because of the chemistry of the molecule and the ability of silica to interact with those molecules. The affinity is so high that the compound or the analyte can actually stick permanently to the silica, and then it becomes, the column becomes a waste column. You cannot remove it then. You cannot use that column, and these columns are quite expensive, sometimes in lakhs. So you have to buy these columns spending from say 50,000 to a lakh or something like that. These columns are very expensive columns, and the service engineer also, if your silica, if your column is blocked or if your column is not functioning properly, you have to call a service engineer to see, look at the instrument, and he may charge you a bomb for that. So the maintenance of such things becomes very difficult. So this is all about partition, adsorption, chromatography. I hope you all have understood. Are there any doubts? Yes, are there any doubts? So these adsorption chromatography, partition chromatography, we have studied today. Paper chromatography, we have discussed, we have almost actually discussed high pressure liquid chromatography, but I will tell you all the, show you the working of it. Okay, and affinity. So these are some things we have yet to discuss. Affinity chromatography, ion exchange chromatography, thin layer chromatography. Adsorption आज हमने किया है, so it should be easy for you. And gas chromatography is removed. So we will discuss all these types, and also I'll go through your syllabus and see what is there in the syllabus. So in the next class, we will do affinity chromatography. Do you have another class after this? And do you have this 8 to 9:30 every day? Yes, do you have? Yes or no? I didn't get it.
No, ma'am.